Hey guys, in this video we are going to see a type of passive transport, diffusion. So here I have drawn a cell and those blue dots indicates a solute which is present inside as well as outside the cell. So diffusion is a passive process. That means it doesn't require ATP for energy. So diffusion is basically movement of solutes from high concentration to low concentration. So if you could see in this picture, so obviously the solute movement should be from inside to outside along the concentration gradient. So when it comes to diffusion, there are two types. The first one is simple diffusion and the second type is facilitated diffusion. So simple diffusion is what I've described. It's a movement of solute along the concentration gradient. So whereas what is facilitated diffusion and how is it different from the simple diffusion process? So here I'm drawing a cell with solute concentration inside and outside the cell. So if you see here, according to the diffusion process, along the concentration gradient, if the solute has to move from outside to inside the cell. But here you require the help of a protein called carrier protein that is present in the cell membrane. So this protein facilitates the transport of solutes along the concentration gradient. So for facilitated transport, you require the presence of carrier protein on the cell membrane. But the disadvantage of this is the saturation of carrier protein. So when the carrier protein is saturated, it is going to halt or it is going to stop this process of transport, diffusion process. So one of the examples for carrier protein in the body is glucose transporters. Those are called as GLUTs. So to be very specific, one example that is related to this carrier protein is SGLT2, sodium glucose transporter 2 in the kidney. So this is one of the carrier protein present in the nephron epithelial cells, tubular epithelial cells, where this particular carrier protein helps in reabsorption of glucose from the luminal fluid into the epithelial cells, from there to the circulation. So if you see, out of the glucose that is filtered, 100% glucose is usually reabsorbed with the help of the proteins present there. But imagine in diabetes mellitus, when there is excess blood glucose levels, when all this glucose is filtered in the glomerulus, this excess amount of glucose is going to saturate this carrier protein. So in that case, when the carrier protein SGLT2 is saturated, the glucose cannot be reabsorbed. So this excess glucose is going to be excreted in the urine. So that would lead to presence of glucose in the urine, and that is called glucosuria. This is seen in diabetes mellitus, and one important thing is the saturation of carrier protein SGLT2 in this condition. So now to represent this diffusion process in a graph. So in this graph, in x-axis, the concentration gradient or the concentration of the solute is shown, and in y-axis, it is the rate of diffusion. So two graphs I will draw. The first one in the dotted lines is for simple diffusion. As the concentration increases, the rate of diffusion increases. Whereas if you see the blue graph, which is for facilitated diffusion, initially there is increase in rate of diffusion as there is increase in concentration, but after a point, even if concentration increases, the rate of diffusion will not increase. It will reach a plateau, and the reason is saturation of carrier protein. So to recap, diffusion is basically movement of solutes from high concentration to low concentration along the concentration gradient. There are two types. So simple and facilitated. So facilitated diffusion is the process which requires the presence of carrier protein in the cell membrane. But the major disadvantage of this process is saturation of carrier protein. So one such example is SGLT2 in the kidney, And in diabetes, saturation of SGLT2 results in glucosuria. So if you see the graph, the simple diffusion graph is exponential, whereas the facilitated diffusion graph will show the